All right, welcome to Evolution Podcast 6. We are still compiling all the evidence that evolution has actually occurred, that species have changed on Earth over time. And our next little packet of evidence is called comparative embryology. So let's get right to it. Remember that the evidence for evolution comes from 5 major areas of science. And none of these has to stand alone by itself. They all weave together to make this incredible mountain of evidence that supports evolution. So as far as comparative embryology is concerned, what is it? Simply what it sounds like. It is the study and comparison of the embryonic development and the structures of different kinds of species, comparing them to each other. What we find when we do that is that closely related organisms, like maybe a mammal group, for example, they go through similar stages in their embryonic development. And the more closely related you are, the more similar the stages are that the organisms pass through as they develop. So, for example, all vertebrate embryos, all of them, fish, amphibians, reptiles, birds, mammals, all of them go through a stage in their development in which they have gill pouches on the sides of their throats. As we can see here, this is a fish, a reptile, a bird, and a human embryo. And highlighted in this purple color are these gill structures, these very primitive little pouches that form in every single one of these. Obviously, fish are the only ones that later grow up to actually have gills, whereas all the rest of the organisms do not. So why would they all start out with these fundamental little pouches in the same basic positions on their bodies? The idea is that common ancestry is probably the answer. So why do embryos of very different species have structures that are almost identical early in their development? It suggests common ancestry. Okay, sorry that I said that before we actually got to the slide. So at the earliest stages of development, similarities between, for example, fish, frogs, snakes, birds, humans, all vertebrates is much more evident than the differences between them. Obviously, differences become more pronounced later in development, but initially they look really similar. So not until development progresses that those original structures slowly diverge and take on the obvious characteristics of each different class of vertebrate. So comparative embryology supports evolutionary ideas in a number of ways, but in a key way, what it does is it reveals homologies, homologous structures that are not necessarily really visible in adult forms. Things look very different in the adult organisms, perhaps, but if we look back in the development of the embryo, we can see that those structures really do start out looking very much the same. So what we see here, we have a fish, amphibian, reptile, bird, and then we've got 4 mammals over here: pig, calf, rabbit, human. And what we see is that initially, if you look at all these embryos early in their development, in this top row, there's a whole lot of similarities. Unless you were an expert, it would be really hard to tell the differences between them. And then as development progresses a little further, you start to pretty easily be able to tell the fish from the tortoise or the chick, and the salamander from the tortoise or the chick, are all these mammals. But if you look at these more closely related mammals, they have a lot of similarities. And again, if you were not an expert, you might not be able to tell apart. And then even later in development, we can see some really obvious differences. Still more similarities over here, but differences starting to come up. But what we see is those that are more closely related have more in common for longer in their development than organisms that are less closely related. These guys have more in common with each other than they do with these guys that they are less closely related to. Okay, so just to reinforce, fish, those gill patches are going to develop into gills, but in the terrestrial land-based vertebrates, those gill pouches become other things. For example, in humans, those gill pouches become parts of the inner ear structure, eustachian tubes that connect the middle ear with the throat. So comparative embryology is able to establish homology in structures where that might not be so apparent in the adult forms. Okay, and that's just a picture of a human embryo and those gill structures. Okay, and that's it. We'll pick up with the very last podcast in this discussion of evidence for evolution in the next podcast. So see you then. Take care.